Hey, what's up YouTube? So in this video I'm going to talk about Unit 2.1 of the IB Biology curriculum, which basically covers cell theory. What I've done here is made notes which I've derived from a variety of different IB Biology textbooks, and I've just written all the important things that we need to know for the final paper, nothing more, nothing less. And so let's just get into it. First one is Unit 2, Learning Outcome 2.1.1, which is just basically outline cell theory. And that's pretty straightforward. All organisms are composed of one or more cells. Cells are the smallest units of life, and all cells come from pre-existing cells. It's pretty straightforward. I mean, you just got to memorize it. The second part is discuss the evidence for the cell theory. And so basically for the first part of the cell theory, it's just various scientists through the use of tools such as a microscope have observed small independent beings. And that's a quote from a scientist, which I haven't really bothered to remember because I don't think you need to. I think if you just say through a microscope, it should be fine. For the second point, thus far, they haven't found a single entity that hasn't been made of at least, or living entity that hasn't been made of at least one cell. So that gives evidence that it's the smallest unit of life. And thirdly, all cells come from pre-existing cells. Well, experiments have demonstrated that living things do not spontaneously reappear, but they only can reappear after being exposed to pre-existing cells. I think the example was they put two different chickens in two different buckets and basically exposed one to pre-existing or living thing, and then that one made more cells, something like that. I don't think you really need to know. If you say these three things, I think should be fine. Third one is just a state, not really going to worry about it. The fourth one is compare the relative sizes of molecules, cell membrane thickness, viruses, bacteria, organelles, and cells using the appropriate SI unit. So this one's really just about memorization. I think it's easy to organize it in the way that's been done here, 110.1, 110.1, micrometer, then to nanometer. So basically start off with a cell, which is your basic which is your basic entity that you want to talk about. When what's in a cell, it's organelles. Bacteria, virus are can be basically classified in the same category, so they're in the middle. And then your membrane thickness and molecule both start with M, so you can just kind of put them at the end. Something like that. I mean, it's up to you however you want to do it. I've memorized it this way. For 2.1.5, it just says basically explain how to calculate the linear magnification. I think all you really need to know here is the formula for it, which is basically magnification equals size of image over size of specimen. So what does that mean? Basically, when you look through a microscope, you're going to have an image and you're going to have a magnified image. So it's going to be, if you're looking at a planaria, it's going to be the size of a big version of a planaria. So you're going to measure that with a ruler. The size of the specimen will probably be given to you, and therefore you can just simple division and you'll get the actual size. So if, let's say, your actual size of your image is 10 centimeters of the planaria that you're looking at, then, if that's the one that's on the paper, and then the actual size like 10 nanometers, all you do is 100, you convert them both to the same unit and then you divide them. Pretty straightforward. The surface area to volume ratio was at first for me a little confusing. It says explain the importance of surface area to volume ratio as a factor for limiting cell size. So basically the key principle that you need to know is as the size of a cell increases, the surface area to volume ratio decreases. So what does that mean? Well, basically the surface area will increase, but it will increase at a much slower rate when compared to the volume. And there are some formulas in a textbook that explain how that happened. You don't need to know that. All you need to know is that the surface area will increase, but at a much slower rate when compared to the volume. So the size of the cell obviously affects the rate of its chemical reactions, because the bigger the cell, the more chemical reactions that will occur, the more waste that will be produced. So the surface area is mandatory for a cell because it allows for molecules to move in and out of the cell. So if you have more of a volume or more of a size of a cell, but not enough surface area to compensate for all the waste that needs to be removed, then you're not gonna have a very efficient cell, and it won't be able to carry out all the functions of life. And therefore, the surface area to volume ratio limits the cell size, because the cell needs to stay at a size where it's optimal for the cell to operate. So that's basically how the surface area to volume ratio limits the cell size. 2.1.7 is just a state question, pretty straightforward. Then 2.1.8 is basically asking to explain that cells in multicellular organisms differentiate to carry out specialized functions by expressing some of their genes and not others. So basically, the given in that question is the definition of differentiation. So the first thing, you can just start off with a general point. Every cell in a multicellular organism contains all the genes of that organism. So it will always have all the genes. Question is, which genes will be turned on and which genes will be turned off? And that's what happens when a cell undergoes differentiation. Some genes, depending on where it is, depending on what pathway it takes, will be expressed, and others won't be. So that depends on the pathway of development. It's a key word. Then once the cell has become specialized, or in other words, once it's chosen its pathway of development, it can't change, it can't go back. That's how it's been specialized, that's how it's going to stay. So an example of that is the gene for keratin, which is a protein. It will become active in your hair and nails, because that's what makes up your hair and nails. So therefore, that's just an example of specialization. So 2.1.9 is just a state question, and then 2.1.10 is basically give an example of stem cells. Stem cells are basically undifferentiated cells, so they haven't been specialized yet. So they can essentially be, like for instance, if you have Alzheimer's and you've lost if you've lost neurons, then they're trying to get stem cells to differentiate into neurons so that they can compensate for the lost neurons. But a much easier example is simply bone marrow transplant. If you have cancer and you're going to undergo chemotherapy, the chemotherapy can't distinguish between which cells it's going to kill. So therefore, it will just kill all of them. So in your bone marrow, there are stem cells. And basically what doctors will do is they'll harvest those stem cells out of your bone marrow using a needle. Then they'll do the chemotherapy on the patient, and all the cells will die. And then what you can do afterward is you can put the stem cells back in through your vein. The stem cells will find the bone marrow, and then they'll start making the blood cells again. And so basically you're back to what you should be. And that's basically Unit 2.1 which is covering cell theory. Feel free to, if you want to pause the video at any time, to just copy down notes, or hopefully this is helpful for you guys. And thanks for watching.